Okay, can go ahead and get started. Thank you.
All right, well, welcome everyone to this 3D electron microscopy, kind of a review and introduction to an instrument called the VolumeScope. It's presented today by Tara Niles of ThermoFisher Scientific. This instrument is very interesting. The EM Core has had its eye on this instrument for a couple of years. We've sent some test samples and had them evaluated. And so we thought it would be a good time to share what this instrument is with all of you and get some feedback and let you know that this instrument is available and this, well, not available for us yet, but we would like, the technology exists. And so we want to know, will this technology work for you here at UF? And if so, you can follow up with the EM Core and provide your insight and feedback so that we can see if we're going to move forward with this and what our next webinar might look like. I'm going to hand this over now to Tara.
Wonderful. Thank you very much, Karen. And when Karen and the rest of the organizing team asked us to talk about 3D electron microscopy, I was so excited because for many, many years, I've been in traditional electron microscopy, and I believe many of you in the core are in electron microscopy as well. And when I was exposed to the world of 3D electron microscopy at a level in the VolumeScope over the last few years, it was so exciting because it truly opens up a new paradigm in electron microscopy. So I'm happy to share that with all of you today. And as we get started, I'll talk a little bit about the rationale behind why we want to consider 3D electron microscopy. And as many of you may know, by working with 2D electron microscopy or even 2D imaging itself, you know, we're used to looking at sort of a 2D image, a projection on a screen, or even a picture on our iPhones and our cameras. And when we look at that, we realize in our brains that that's actually a representation of the actual 3D object that we're taking a picture of. But our brains have to do sort of that interpretation. And when we do that, we might be missing something based on what our current schema is. So if we look at the cartoon now, we could see that the projection is not always obviously what the 3D object actually is. And so by looking at the direct 3D object itself, we truly get the real representation. And I'll use something a little bit more, an example that's a little bit more biologically relevant to us now. We could see the tubule here in this graphic, and we could see that it is, of course, a 3D object that has, you know, a circular 2-dimension, but also it has a 3-dimension that kind of turns and curves and folds back on itself. And whenever we take a cross-section or a 2D image of a certain area, it may be misleading or incredibly complex for us to figure out what it's actually representing in 3D space. So on the very top 2D projection, that's easy. You know, we look at the cross-section and it's a circle and we're like, aha, okay, that's, you know, that's a tubule, but we don't know if it's, you know, if it's a straight tubule and so on. And then when we take different 2D cross sections through different planes of the 3D structure, we get all of these varieties of projections, and it becomes really challenging for our brains to make that interpretation of what we're actually looking at. And so this is why we want to look at 3D electron microscopy. In today's presentation, I'll show a few examples of why 3D electron microscopy is used and how it complements existing and other techniques. And we'll spend just a few slides on the workflow and then wrap up with some application examples of 3D electron microscopy. So the science and the research that's being done that has helped people solve problems. The first example that we're looking at here are mitochondria. And of course, this is classic in life sciences. And when we look at the TEM image of mitochondria and bat liver, we certainly see that really traditional, that classic bean-shaped projection, that cross-section in our TEM section. And we could say then make the assumption that all mitochondria are pill-shaped based on that image and our prior knowledge of it. But we could also see that there are other possibilities. And so depending upon our perspective on the mitochondria, we could actually see that they are in fact more complex, like the bifurcated area on the right and how much it branches out from the initial area. And so with 3D electron microscopy, what we're able to do is collect a volume of material samples, and that's the name for the VolumeScope. And when we collect that volume or sequential sections of images, we could then render them into 3D space. And then we render that block or volume rendering. And we could select certain features. So for example, if we wanted to select only the mitochondria, we would segment them in blue using a data visualization package. And then we can keep sectioning through visually through the image analysis package so that we remove all the material that we're not interested in. And in this example done for Temple University, we're able to segment out the mitochondria and then do even more investigations of them, like how much is the volume fraction of that? So how much of the total tissue space do the mitochondria occupy? And then we could even do individual mitochondria themselves. We can color unique mitochondria. We could also see how many mitochondria are touching each other. And so are they spreading out or are they in fact individual? And then we could do charts and graphs and quantitative measurements to better understand what the actual tissue ultrastructure is doing when we look at that larger volume area. And one more comment on this particular work. There have been now many studies on mitochondria and in particular cardiac tissue. And it is a mechanism for understanding any disease state associated in the heart tissue. And so changes in the mitochondria volume itself on an individual one. There is I'll use the word puffed up appearance. That was, I remember when the scientist, she actually used that word. In the diseased cardiac tissue, the mitochondria are puffed up or inflamed, slightly larger. So each mitochondria has a larger volume fraction. And when you compare the diseased tissue to the healthy tissue, you can see that there's a small difference in volume fraction for individual mitochondria as well. And, you know, of course, that makes sense. Inflamed mitochondria, they're working overtime to help compensate for the cardiac muscle. Another area why we want to use 3D EM is because the electron microscopy aspect of it correlates with light microscopy. And in this really cool example done over at MIT that's accessed by the link there, you could see they were using two-photon microscopy to trigger and register where the receptors were being fired within the dendrites itself. So this was a mouse with the microscope on it in live time, and the mouse was exposed to a stimulus. I think it was a flashing light. And each time the stimulus fired, the response in the brain was measured. And you could do that with light microscopy to see in live time how the response is happening. However, with light microscopy, you don't get the same level of information or detail that you do with electron microscopy. So taking it one step further and excising that area allows the researchers to figure out what's going on at the morphology level of the dendrite itself. And you could see that there are a greater number of structural details, what's actually changing and how does the restructuring work. And another example of where 3D electron microscopy complements other work is when you're looking at quantitative variation. So say in between a wild type and a modified experiment, a knockout or knockdown experiment. In this example here, the norexin heparin sulfate, complex is shown to play a role in neurological and behavior disorders like schizophrenia. And so this was genetically tested. And so we can see that in the wild type, there are varying amounts of these mossy fibers, or MF, and thorny excretions. They describe the type of dendritic growth. And so the fibers, or the mossy fibers and the thorny fibers, are showing that in the wild type, they are much greater compared to the knockout mouse model. However, the charts and graphs themselves don't really tell us all that much. It says, okay, there's less of these dendrites, but what does that mean? Like, what does it look like? And so now when we look at the visual or the graphic, and this is the actual data that was collected from these dendrites on the wild type and the knockout, we could see that in both the mossy fibers and thorny ones on top and bottom, on the wild type on the left, there is more of them, so more of the growth, and the growths themselves are even more complex, so a little bit more branched out. And then if we look at the modified animal on the right, we see that these dendrites are not growing and interacting as much as they are in a normal situation. And that is also work done with the VolumeScope. And so then this allows us to deepen our understanding of relationships between features in cellular ultrastructure and across tissue scales as well. So we could then see where the axon interacts with other neurons, for example, how the dendrite growth happens, where it interacts with other neurons, and then even greater details like the synaptic vesicles in the dendrites and how expansive they are, how large of an area they occupy when we get into the fine details. And all of this builds off of the traditional view from both TEM and FibSEM work. So these are techniques that have been around for a very, very long time. The VolumeScope, which is the data that I just showed you, is a newer technique. And when we look at the TEM images on the top right in this example, we could see that through different magnification levels, we're looking at the intercellular regions and these vesicles or buds that seem to be sort of floating in that intercellular space. So in the TEM, we've got the great luxury of zooming in further so we could see greater detail. And we could even zoom in on one area where these vesicles, so to speak, appear to be budding off of the parent cell. And so again, back to that user interpretation requirement, we might infer that those are budding off and then floating in the space there. But then when we incorporate a 3D view to it, such as with the FibSEM that we see here, we could actually trace all of those areas throughout deeper and deeper within the sample itself. And as mentioned in the data reconstruction capability, we can colorize each of the quote parent cells in different colors. So the orange and blue, completely random, orange and blue. But we could see that those vesicles, which appear to be separate and distinct from the parent cells, they are in fact at some point in the volumetric space touching the parent cell. And when we move over to image F on the lower right-hand side, we could see that all of those, they're actually fingers that are coming, up into the cross-section that's on the top 2D viewing plane, but they do actually interact and touch with the parent cell at some level lower or deeper in. And so the concept has been around for a while with FibSEM, but we've opened it up with a greater tissue exploration level with the VolumeScope. And that brings us to an overview of the different aspects of workflows in electron microscopy and for the ones in large volume analysis. So you may be familiar with cryo-EM or single particle analysis, which works at the protein at the smallest level, of atomic level resolution. And we could even use that in tomography so that we get some contextual view of how the proteins interact with the cellular components. But when we move over to large volume analysis on the right, we're able to actually visualize the larger volume of ultrastructure and look at the context of how each component interacts with other components in the tissue. And in the world of VolumeScope, we're actually looking at much larger volume. And this builds off of classical microtomy, where we use physical sectioning to section off materials, thin layers in a block. And so if any of you are familiar with sectioning or SEM or TEM sections, even obviously light microscopy, you know that there are some challenges associated with that. It's a very user-intense technique. And so serial Blockface allows some changes that make it a whole lot easier for users to obtain very, very large areas of tissue while still achieving higher resolution of electron microscopy. And so in these two areas to the left-hand side, we could see the large volume, the anatomy almost of the zebrafish embryo, and then we could zoom in and see a little bit more detail of the retina. And then by the time we get to other EM techniques, we're looking at very, very small areas. Now, of course, the file size is very large because we're collecting many, many 2D images and then putting them together into truly a data block. And we'll get into some of that. And so large volume analysis incorporates several different techniques. So if you've heard of the term large volume analysis or LVA, it usually includes one of these. The most traditional one is FibSem, and if I was with all of you guys in the room, I would ask everybody to raise their hands and say who has heard of FibSem or used FibSem, and usually we get like 75% of people around who have either heard it or used it themselves. So FibSem has been around for a while and proven valuable as a technique. And of course, I could also ask who has done traditional microtomy, prepared thin sections on tape or film. And many people have also done that. Serial Blockface actually brings those two techniques together, the EM component and the microtomy techniques together by doing in situ sectioning. So preparing the block and then popping it into the SEM. And the way that it differs from array tomography is because you cut each section and you place it down before you put it into the SEM, and then you're able to image section after section in sequence on a wafer or plate. And FibSem uses an ion beam to actually do the milling or the cutting away. So it uses the ion beam to actually cut through the material and disperse the material inside of the SEM chamber. And serial Blockface imaging in the middle actually uses a block inside the SEM and a physical microtome or a diamond knife inside of the SEM that continually cuts away the surface. Now, one of the things to keep in mind is how it's done, and I'll show you a little bit more on how it's done in a moment. The main components with the hardware are the base SEM. And so this is usually where it gets to be like a little bit hard to conceptualize, like the light bulb starts going on. But it actually truly is an SEM with a microtome inside. And so we use the word in situ microtome to kind of get it to sink in. So inside of the SEM, which is the imaging, we have a specially designed microtome to section away the material. And for those of you familiar with EM, you might be saying, oh my gosh, you're actually cutting resin inside of the SEM. And you're right, you would be correct to be worried about that, because those sections, after we use them, they have to go somewhere, and we don't want them to go up in the pole piece or down on the sample to ruin our next image. So what we see here by this red plus is we have a debris trap that has an electrostatic charge to attract the cut section after we're done using it. it actually becomes debris or unusable garbage after we've imaged it the first time. So that differs from traditional array tomography. And then we have various other hardware and software components that all tie together to do the 3D collection so that it's easy to use, that any SEM operator who's familiar with data collection can just simply set up your region of interest, define your parameters, and do the collection. And then finally pair it with a data reconstruction package to do your volume rendering or your 3D investigations to do quantitative measurements and to, again, say, figure out what your volume fraction of mitochondria in cardiac tissue is, and so on. And then, of course, this is where the high data storage comes in place. So we do have hardware set up to support this. So additional data servers and collection capabilities so that all of your data is preserved. And in this short video, we show some of the highlights of how we swap from a traditional SEM and the Embryo VolumeScope. The Embryo is a field emission instrument, a high resolution instrument. So we remove the main stage and then slide the microtome assembly onto the main stage and then twist some of the brass screws to fix it to the stage, and then, of course, feed through to help disable certain functions of the regular SEM to prevent any disasters from happening. So this is where the VolumeScope is well designed so that, for example, you don't use immersion mode when you have your diamond knife in there, or you also don't rotate or get too close to the pole piece so that you avoid any damages. And then this is the specially designed low vac holder for the VolumeScope, so a little bit different. We have a voltage ring that helps bias the detection so that it prevents charging or reduces charging. And then here's where we attach kind of a traditional diatome knife, adapted for the microtome assembly physically, but same old kind of diamond knife. And then simply screwing it on. And then there's factors like the approach and lining it up, lining the sample block that you see here right in the middle. So this little dark black area right here, if you could see my screen, that's the actual specimen itself, usually a millimeter or so. And then we use cameras to get a rough location, bringing the sample up to the knife so that you don't cut too much nor too little before you actually close the chamber door. And then an overview of the process and procedure. So this falls into techniques that we call serial techniques or serial blockface, which means we image and then cut and then image and then cut and image and cut, and we do it over and over again. And the real difference here between other techniques is traditional microtome techniques is that we're actually imaging the block face itself, and then we cut off that surface anywhere from, say, 20 to 50 nanometers thick of a surface. And so we cut off that surface, and then we image the surface below it. And then we cut off that surface, and we image below it. And so we end up with a stack of cards, so to speak, a stack of 2D images from which we render the 3D information. So the main point here is that we're imaging the block face. So you don't actually have to worry about cutting artifacts or the sections. You don't have to worry about the sections curling up or getting wrinkles in them from dragging of the knife or anything. Some of that on the block face, but largely we've taken care of that. And once the area is imaged, then it becomes, as we cut it off, it becomes debris. So you don't actually have to worry about placing it anywhere, and it gets captured with the debris trap. It means that it can be done sequentially and automated. So a user just sets it up and then goes away overnight. And you may have seen in some of the previous examples, we could cut anywhere from hundreds to thousands of sections over many microns in distance. And so it is actually kind of cool and fun to sit there in the lab and see each section as it's collected. But when you do the many hour runs, then you're free to go outside or enjoy a cup of coffee and even monitor the collection from a remote location. Okay, and we'll have our questions and answers at the end of the session. So I just want to wrap up with a few scientific examples of where the VolumeScope and related techniques are being used to deepen understandings in science. And so what we're looking at here is a work on the Brain Research Initiative through NIH. And Vanessa and one of our users over at UMass Amherst have been partnering together, submitting some samples to our nanoport to collect some data, and all of this supplements the work that they're doing with the NIH on the Brain Initiative. And so in this work here, they're looking at correlative light and electron microscopy to understand various levels between animal organism level through to the cellular and subcellular level. And most interestingly, they're even using light sheet microscopy to incorporate that behavioral aspect. So they were using microelectrodes to study the neuron connections. And like when they stimulate a certain area, does the nudibranch, does the sea slugs turn right, does it turn left? And so by mapping that with light sheet microscopy, they sync in together the different areas of the neural ganglia, and then understand the behavioral response. But of course, behavioral is just one aspect of it. What's the physiological differences that are going on at the cellular and tissue level so that we could understand where those connections are happening and how the behavior leads to change in the cellular area. So a region of interest was collected from the ganglia area, focusing on the pedal ganglia, and then the higher resolution image of a horizontal field width of 242 microns was collected. And now we could look at the 3D movie, where at first we could see how the pedal ganglia attaches and detaches on the top there. So it is just attached by ganglia, the ganglia is just attached by that stalk. And then we could zoom in for a higher resolution view. And what's notable here is in the center, there's a distinct lack of cell bodies. And so sort of dense material, but not very many cell bodies. And then so the collection area and the region of interest zoom in on the video, the movie file that was created, now looks at some of these vesicles closer to the outer membrane. And this whole area was collected, but as you do the visualization, you could zoom in and zoom out. You could retain the higher resolution of the original collection. You could focus in on a certain area to look at that area in detail. And of course, rotate it, select certain areas by color or by other morphology. You can do some intensive work on segmentation of looking at specific features of interest and selecting those and segmenting those out. And then you could, of course, cut through them and see different perspectives as well. Now to show an example of a high resolution capability of the collection. So this is a mouse retina that was done really pushing the limits of resolution for pixel size. So this has a 5 nanometer pixel, and the voxel size or 3D pixel is 5 by 5 in X and Y, and then 40 nanometers in Z. We could actually even go deeper than that or finer than that by using a technique called multi-energy deconvolution. I'll show that in a video just at the end of the presentation. And so by doing this, you're actually able to see fine resolution detail. Oh, I'm sorry. Unless you squint now. Normally I have a zoom in, but it looks like that didn't activate. So zooming in onto the area, you could actually see the ribbon synapses that are really down at the 5 to 10 nanometers size for those features themselves. And the one of interest, I think, is right here in the middle. And then coming back out to a last example where we're looking at a zebrafish embryo, we can actually see the entire head. And so this gives you an idea of the scale and the context. So as we slice through, we could see the embryo, the eyes, and then the other features in the head. And here's an example where the slices were at 829 slices for this one. The slices are much larger, so 100 nanometers or thicker slices, and this allows you to cover much greater space, especially compared to something like FibSem, which would only be a few nanometers in thickness for each of the Z stack thickness slices. And so ultimately the volume that's covered is 350 by 350 microns, which is pretty, quite large, but also keeping some of the detail that we could see here on the back side of it. And another comment is that low vac capability also works when we're using VolumeScope. So even if you have sample with a high amount of resin or a high number of vacuoles, then we can still use techniques to help minimize the charge and improve the image signal that we see. And of course, we're working at low beam current and low beam energy as well that go hand in hand with the biological materials. Okay, so now I will open it up to questions. I have about a 2.5-minute video that I figured I would play after the presentation and during the Q&A section because it shows some of the technology side. So for anybody who's a little bit geeky and really curious about how the technique works step by step, we'll play that video while we ask questions and answers. Before I go to that, though, I just would like to draw your attention to a QR code that we have on the left-hand side. This is for an upcoming Introduction to Cryo-EM webinar that we're holding Thursday at 2 o'clock. And this is kind of like an everything that you wanted to know about cryo-EM, but were afraid to ask. It's sort of a very intro to cryo-EM presentation. So all you have to do is hold your activate your camera and hold your phone up, and then that QR code should get you right to the registration link. And I'll also send it to Karen and Chris and the organizers afterwards if you're interested. And there's my name and email address. So if you'd like to send me an email offline, please feel free to do so. And also you could ask Karen or anybody else to get a hold of me. Okay, and I think there was a chat, so I'm just going to open that up. Absolutely, yeah. I'll send you the slides afterwards. Please feel free to use them in any way that might further educate your group or be useful. It's a really exciting technique. It does require some of that, leap of, change of thinking. So yeah, hopefully some of the videos will help with that, and we will send you that afterwards. Okay, so as the video plays, I will ask if there are any questions that somebody would like to ask either via chat or audio. If you have a question, just make sure you unmute yourself and then go ahead and ask away. So this video shows what's going on inside of the SEM chamber. There's your sample block and then your microtome. And the block is lowered and the microtome approaches over it, removes the top section, and then one of various detectors are used to detect the signal. Here's that multi-energy deconvolution, which actually uses a number of different beam energies to sequentially go deeper into the sample to generate an image further below the surface. And that allows us to get more sections of a smaller Z height without doing as many physical sections, which can be a challenge to do very thin Z sections. 15, 20 nanometers is achievable, but you need just perfect sample prep. And then so here's the sequence again. So another layer is removed, so physically removed. And then that series of images using that multi-energy deconvolution is collected and detected by the in-column Trinity T1 detector, which gives you a view of backscatter and contrast. And we do an inversion typically because the heavy metal stain. And then here's an example of the 3D block and the data that you achieve and the work that you can use using a reconstruction package. Okay, so I'll stop the recording now in case there's anybody who would like to ask a question and not be recorded. I'm doing so. Recording is now